For decades, scientists largely overlooked fat in the brain, assuming it played little role in neurodegenerative diseases. Plaques and tangles dominated the narrative, taking all the blame for cognitive decline and Alzheimer's progression. But Purdue University researchers are challenging that long-held assumption, uncovering a remarkable story in which fat itself becomes a key player, and one that can be managed, reversed, and potentially used as a therapeutic target. Their study, published in Immunity, demonstrates that the accumulation of fat within microglia, the brain's dedicated immune cells, weakens these cells' disease-fighting capabilities. Rather than being passive bystanders, lipid-laden microglia are central to how the brain responds to damage, and understanding their behavior opens new avenues for preserving neuronal health and combating Alzheimer's. Microglia are the brain's janitors and personal trainers rolled into one. They patrol the neural landscape, clear misfolded proteins like amyloid beta and tau, and regulate inflammation to keep neurons functioning properly. Yet in Alzheimer's, these cells begin storing fat excessively, forming lipid droplets that impair their mobility and efficiency. The result is a slowdown in the brain's natural defenses. The very cells designed to protect neurons are hindered by their own metabolic burden. The Purdue team, led by Gaurav Chopra alongside graduate students Palak Manchanda and Priya Prakash, sought to understand why this happens and how it might be corrected. Earlier work by Chopra's group highlighted that astrocytes, supportive cells for neurons, release fatty acids that can become toxic under disease conditions. Collaborations with the University of Pennsylvania connected age-related mitochondrial dysfunction in neurons to fat accumulation in glial cells, signaling a metabolic dimension to neurodegeneration that had been largely ignored. These findings together suggested that targeting plaques alone is insufficient. We're finding that reducing accumulation of fat in the diseased brain is the key, Chopra explained. Accumulated fat makes it harder for the immune system to do its job and maintain balance. By targeting these pathways, we can restore the ability of immune cells like microglia to fight disease and keep the brain in balance, which is what they're meant to do. The team's work builds on a century-old observation. Alois Alzheimer noted that brains affected by the disease contained not only plaques and tangles, but also cells packed with lipid droplets. For years, those lipid deposits were dismissed as irrelevant byproducts. Chopra's research, however, provides strong evidence that these fat accumulations, now termed lipid plaques, are more than incidental. The problem isn't the droplets themselves, but their accumulation. The composition of the lipid molecules within microglia and astrocytes appears to drive neuroinflammation, contributing to Alzheimer's and other age-related pathologies. Essentially, lipid overload transforms microglia from vigilant defenders into metabolically encumbered cells, reducing their capacity to clear harmful proteins and maintain neuronal health. The Purdue study zoomed in on microglia near amyloid beta plaques and revealed striking findings. Microglia located closest to these plaques contained twice as many lipid droplets as those farther away, and they cleared roughly 40% less amyloid beta. Further investigation revealed that these cells, when in contact with plaques, convert free fatty acids into triacylglycerol in quantities so large that they become immobilized by their own fat. Central to this process is the enzyme DGAT2, which catalyzes the final step in fat storage. Interestingly, DGAT2 was not overproduced at the genetic level. It simply accumulated because it was not degrading as quickly as it should. This bottleneck effectively forced microglia into long-term fat storage instead of energy use, leaving them less capable of performing their immune functions. The remarkable and optimistic discovery is that this dysfunction is reversible. By either blocking DGAT2's activity or promoting its degradation, researchers were able to reduce fat accumulation, restore microglial activity, and improve their ability to clear amyloid beta in animal models. In other words, these cells are not permanently damaged, they are temporarily overwhelmed. With targeted intervention, microglia can regain their efficiency, resume their cleanup duties, and maintain a healthier brain environment. This finding shifts the focus from treating plaques directly to supporting the brain's natural immune system, offering a strategy that empowers cells to fight disease on their own terms. The implications are profound. Fat accumulation in microglia is not only relevant to Alzheimer's, but is also observed during normal aging and other neurodegenerative conditions. Therapeutic strategies that restore metabolic balance in microglia could therefore protect cognition more broadly, slowing the effects of aging while enhancing the brain's resilience. This perspective reframes Alzheimer's from an inevitable, plaque-dominated decline to a condition where intervention is possible, where the brain's own defenses can be revitalized, and where cellular recovery offers genuine hope. In essence, Purdue's work paints a new picture of neurodegeneration, one in which fat-laden microglia are both villains and heroes. They become a problem when metabolic overload slows them down, but they also hold the key to recovery once that burden is relieved. By restoring microglial function through targeted lipid management, we can help the brain fight disease naturally, preserve neuronal health, and maintain cognitive balance. It's a story of resilience, possibility, and the remarkable adaptability of our own cells. A reminder that even in the presence of Alzheimer's, the brain has tools to defend itself, and science is beginning to show us exactly how to unlock them.